All right. Hi,
Hello.
Ben.
Hi.
Finally. Hi. All right. So we'll get started. Hi, everyone. Thank you for joining us and welcome to the 14th Shiny Gathering. So today is a very special one because we are doing it in collaboration with Pharmaverse and we have Ben with us today. So, hi, I'm Gigi. I'm the internal journalist at Epsilon, and Ben is the principal programmer at GSK, as well as the author and maintainer of the Admiral package, which we'll be talking about today. So he will be sharing with us everything we need to know about the package and how to make the most of it for creating ADaM datasets. So please leave your comments in the chat as we'll be taking them towards the end of the session. And now I'll hand over to Ben to introduce himself to us and tell us more about Admiral.
Yeah, thanks, Gigi, and thanks, Epsilon, for putting this together. I'm gonna try to share my screen. So window. Over there. All right. So let me render the Corto document and we'll get going. Okay, yeah, so I'm here to talk about the ABRoll R package, and thanks again for Epsilon for putting this together. I appreciate it. And what I'm gonna go over today is just like background about myself and what I do with ABRoll and GSK, where I work at, and then go over to the ABRoll history, kind of what we're trying to achieve with ABRoll, um, and the ways of working behind the scenes, and what we're kind of planning beyond ABRoll. And I'll be jumping around from different tabs, so I apologize. I hope it's not too too jarring, but I didn't really want to copy and paste things into a slide deck, so I kept the slides pretty simple. And then, so that will probably take us into like 15 minutes, the background and overview, ways of working of ABRoll. And then I'll do a little bit of light coding, just showing you how to start up with ABRoll if you're interested in using the R package and some of the conventions that we use in ABRoll. All right. So for background about myself, I'm just going to go over some of my experiences using R and some software development knowledge I've been gaining, and let you guys kind of know what I do at GSK, as well as what I do with ABRoll. Um, I, I'm not the I'm the maintainer of ABRoll, but I'm not the what's a good word, progenitor of ABRoll. That was others who came before me. Um, but yeah, just a little bit about myself. So I have limited experience with software development. This is like my first big software development project working on this ABRoll R package, and it's definitely worthwhile. I definitely encourage folks, if you're interested in getting more into software development projects, to look for open source um, R packages or Python libraries. You can definitely gain a lot of cool knowledge and meet really cool, interesting people. I think the reading of software development and R programming is very important. But when you're able to read it and actually do it on an open source project, I think it can be really worthwhile. And a little bit about me at GSK, just to say I've learned SAS, which is a kind of the staple of programming languages for the work I do at GSK. Um, I've learned SAS at GSK over the last 6 years, and before that, I learned a lot about R in my university experiences. Um, and over the last couple years, I've really noticed a uptick of interest in the section of the pharmaceutical industry that I'm in using R, which I'll go over more. So I've really tried to push myself to learn more deeply about how to use the R language. I highly recommend this book, Advanced R, if you're interested in learning more. So at GSK, the things that I do day to day, I'm a programmer of ADaM datasets, which is sort of um, the the last data set you kind of put together before you start rendering tables, listings, and figures. And ADaMs are part of a larger framework called CDISC, which I'm not going to go into too detail, but it's just a set of standards that really kind of encourages traceability of where the data has come from in the clinical drug trial. And the ADaM datasets, which the ABRoll R package tries to build, um, forms the core of that package, or that submission package, and feeds into these table listings and figures. And I was kind of trying to connect it a little bit to Shiny, is like a Shiny gathering. And I think things are changing in the industry, and I think we're going to see more interactive dashboards, table listings, and figures that are interactive. But the source of those, I think a lot of the sources of those TLFs for drug submissions, are still going to be ADaMs. Also at GSK, I oversee other programmers who are building ADaMs and TLFs. And at GSK, we call those folks, like myself, study lead programmers. So we kind of oversee folks working on the project. And at GSK, I work on a bunch of other R initiatives besides ABRoll. One package I really enjoy is logRx, which is just making logs of what's happened in the R script. The other language I work in, SAS, when you run the code, you natively get a log file. In R, you don't get that. It's just built differently. So logRx tries to bridge that divide there and makes a log of what you've done. Exporter is another R package I get to work on. When you submit to a drug to a regulatory agency, most of the times they require it to be in an XPT file, which is a format that was created using SAS originally. But you can create it in other languages like R. But there's some specific needs that are needed when you make the XPT for the drug submission, especially if you're reading the XPT file in R and in SAS. It's important that those specifications are met in the XPT. So that's what I get to work on in Exporter. And I also get to work on some other stuff within the Pharmaverse initiative, and developing some internal R capabilities. And one other big initiative I just want to highlight is a big issue is, or something I've seen in the industry over the years. There's a lot more interest in R, but regulatory agencies, while they can't say which language to program in, there's not really a lot of groundwork laid for delivering a submission in R. And there's groups within the R consortium. There's a submissions working group that has done some pilots to help kind of lay the groundwork for future R submissions. And Novo Nordisk has also publicly made it available kind of their learnings from their experiences with doing an R-based submission. And I hope that other companies, as they do R-based submissions, come forward, kind of help make that known. So I did, one of my mini tabs, want to bring up, so I'll do R consortium. So just to let you guys know, within the R consortium, there's several working groups. One of them is what I alluded to, the submissions working group. And let me find their homepage. So it's a small little initiative, but I think it's going to have a big impact. But there's been 4 pilots over the last couple of years that have done simple submissions where we've developed TLFs, table listings and figures, in R, and submitted to the FDA. One, I think, of prime interest. So that was pilot 1. Pilot 2 was looking at delivering the same table listing and figures as a Shiny app to the FDA, which received some good feedback. The third, which I'm very proud of, is pilot 3, where we did the same tables, listings, and figures, but also did all the ADaM datasets in R, and we did that using ABRoll, which I'll go into more about the ABRoll R package soon. And pilot 4, which Absalon's actually involved in, is kind of reimagining how we do submissions and maybe using some newish technology like containers or maybe like WebAssembly to deliver submissions. So I think all this stuff is very exciting. You see a lot of industry changes, and there's a really great video that Posit put together on Nova Nordisk's journey to an R-based FDA submission, which I encourage folks to check out. I think it's about a year old now, but I think these learnings are helpful. So that's kind of what I get to work on at GSK amongst like study work and some R initiatives. So within ABRoll, which just to let you guys know, I'm the maintainer of ABRoll, so I definitely don't see myself as the author of ABRoll, but I help maintain the code base, and I help do releases to CRAN for the package. And we actually run bi-weekly meetings for the ABRoll developers. There's probably about 8 to 10 of us who are core developers from all sorts of different pharma and CRO companies. And then there's about 10 other people who kind of sub in and out when they have time with different issues. But within ABRoll, like a lot of my day-to-day is helping to triage issues that come in. I'll go over more how we use issues on GitHub in just a minute. But being able to write an issue like a work order for developers, I think is very helpful. I do a lot of reviewing of pull requests. So a pull request, if you're not familiar with how GitHub works, is basically just a request to change the code, and we have a pretty scripted way on how we do that. And I'll show you guys that in a minute. And then I help to identify the scope of work for future releases. Some of those things could be new functions, new some refactoring of code, maybe some new vignettes that we put together on how to use the package. All right, so ABRoll. So this is the R package for building ADaM datasets, and going to go into why I think R packages are super helpful for the work that we do. Jump into kind of like how we envision users using ABRoll, the structures and ways of working, how we use GitHub. Not really. I think it's cool to see some of the ways we use GitHub to kind of help work collaboratively together across the pharmaceutical industry. So that's what I'll go over with ABRoll. So just to sing the praises of the R package. So you can, at least for ADaM datasets, which are just, I think a lot of the work that can be done to build an ADaM dataset can be done using base R, maybe some tidyverse packages. But I think what's neat about ABRoll is that you can really scope that body of work that you want to do, that you do day to day, that's repetitive, into the package and align it to the CDIS standards, which is what we follow for building the ADaMs. The package framework also really has a lot of powerful features for rendering documentation and supplying examples. I think this is very helpful for people wanting to learn how to program ADaM datasets. Is being able to like download the package and being able to play with the different examples, and we'll see that in a minute. The package framework also has built-in testing features, which I think are really useful, especially for a highly regulated industry like pharma. Being able to build open source products, but build good unit tests for the functions is helpful. And another shout for building something in the open is, and we'll see this when I discuss some of the issue stuff, is we tend to absorb a lot of edge cases where we built these functions where we didn't anticipate some kind of data scenario, and we can absorb those into our unit tests, which I think is very helpful. And the groundwork has really been laid in GitHub for building great R packages with how GitHub is set up for doing collaborative work. And I'll showcase some of that stuff in just a minute. And just give you some history of ABRoll. It's a project that was started by GSK and Roche a few years ago. We we met together and we started blending our specs, our specification files for how we build our ADaM data sets, and we try to align those to CDISC standards as best we could. There was definitely some growing pains with that, but after 2021, we kind of got really rolling, and we did our first release to CRAN, which is where most R packages are stored. There's other package repositories like Bioconductor, but I think for the work that we do for clinical drug submissions, a lot of that stuff is found on CRAN. And just again to say that a lot of the ADaM data sets that we build as statistical clinical programmers, it's a lot of filtering, joining, ordering of information to build these data sets that feed into the tables, listings, and figures. And again, not to disparage ABRoll, but the code, the underlying code is not hard to follow, which I think is very helpful, especially if you're a regulatory person or someone coming up to speed with ABRoll. I think the code is very lightweight and easy to follow. But what's important here, rather than just using BaseR and tidyverse to program your ADaM datasets, is ABRoll is trying to reduce the repetition of the code. Lots of ADaM datasets, which I'll go over into the different flavors of ADaMs in a minute, have similar derivations. You just slightly tweak them here and there. Oh, timed out. Oh, my posit connection timed me out. All right, back to it. Yeah, so I think the ABRoll code helps kind of reduce the repetition of code and kind of helps us absorb edge cases, provide information to users if things are missing, certain variables, certain things look suspicious. And the big part of ABRoll, which I think is very powerful, and you'll see this when we do the code walkthroughs, is it really is focused on modularity, where every function is building like a variable or adding records to the data set, and it really allows you to kind of see how the data set is constructed. Whereas like I think when you do a lot of programming in BaseR, maybe you throw in some tidyverse packages, it's harder, and everyone does that slightly differently. It's harder to kind of pick out what's going on. Whereas I think ABRoll kind of adds, kind of takes out that kind of code complication and lets you just focus on what's happening in the code. And so I'm going to switch a tab in just a minute, but a lot of our core work is done on GitHub, obviously using R. And I did want to showcase kind of how we work on GitHub. So let me go to the ABRoll website. And I think this is pretty standard for a lot of R packages, but we tend to work where we identify issues that we want to work on in the project. So maybe something with just an update to our documentation or an update to how we organize the R package. And we'll have the issue posted there. We'll have some discussion from developers or from the community. For larger issues, like larger discussions, like maybe refactoring, we'll use this discussion tab here to kind of talk about more deeply about how we're going to do things and then make issues out of that discussion. And what I think is really helpful here to see is all that information that we're discussing, all the code changes that we're proposing, all the ideas that are floating around or bugs that we discover, all that's like documented and out here for people to see, which I think just strengthens the power of this package. So one thing I did want to show is in a pull request, which is like a change to the code that we want to see happen in the code base. Just to pull one up really quickly. Let's see what is going to be a good example of. Let's do a simple one. So this is a feature I love about GitHub and has helped us kind of working from multiple different companies have a standard way that we all approach it, is that we do have this kind of verbose checklist that we have developers kind of walk through uh, each piece to make sure that we're following a certain level of standards. Uh, that's a kind of play by play, reminds you of like what we're following, how to deprecate things, how to categorize different functions, so it renders appropriately in our website on down into details. But the thing that's really important is each time we make a code, we make a proposed change to the code, all these actions fire in the background, and some of these, they're you hear them as CI or GitHub Actions. CI being continuous integration. All these things are helping to check that our links are still working, our style of code is still adhered to, spell checks. But the really important ones is the package is rebuilt, and all those unit tests that we've built behind the scenes are all fired, which I think just again, you can see how helpful that is as a developer or a user using this package, that that a change that's happening, is being tested against it, so that maybe if you're a user of the package, aren't going to be impacted negatively. And if you are, we really try to have a really detailed change log of what we're happening and recommended ways to update your code. So the pull requests and the issues are hugely helpful for how we work um, in GitHub. I think I always like showcasing them as a way. And this is stuff I've all learned while working on this project. I didn't know anything about pull requests or Git or GitHub before I came on this project a couple years ago. And I definitely didn't know. I mean, I've heard of GitHub for a long time, but I didn't know anything about this continuous integration stuff, which I think is very neat. And just to give a shout out to all these great people who come from these companies: Roche, Baird, Navidas, Jim, Latoris, and Sitel, all these great people who come in and help with the ABRoll R package. Um, and just a little bit beyond ABRoll, before I go into some of the coding stuff, um, is there's a standard set of ADaMs that go across all different types of disease areas, but sometimes there's certain ADaMs that just don't go as well with other disease areas. So we've built extension packages for ABRoll. One of them, I'll showcase in just a minute. It's called ABRoll Onco, which is oncology, and these are ADaM data sets that are specific to oncology. So here, I'm not going to show anything beyond this web page, but just know that there is a push to build these extension packages that might deal with oncology, um, metabolic issues, obesity, pediatrics. And really, this is about building those ADaMs specific to those disease areas. But we've definitely found that ABRoll can handle most use cases. We also, ABRoll is part of a larger initiative called the Pharmaverse. And the Pharmaverse is really seen as it's a recommended set of packages for doing the clinical programming work that we do, or statistical programming. It's sort of an end-to-end kind of suite of packages for ingesting metadata, to building ADaMs, to making TLFs, to building up your submission to regulatory agencies. So that's that initiative, which I will. Do I have the site open? Is housed on the Pharmaverse GitHub repo, which I'll just quickly show everybody. Sorry again for jumping around. But this just has example sites, or it has a good site of all the packages that we've kind of pulled together that we think are useful for doing the work that we do, ABRoll being one of them. And there is a blog and an example site that goes along. The example site has more end-to-end feel to it, of moving through working with metadata, working with TLFs. And then the blog site, which I just wanted to give a quick shout to, has releases of different packages within the Pharmaverse, some experiences people have had while working on ABRoll, and some other basic things like that. So, Pharmaverse blog post. And I did want to shout out to data is always a big issue. So if you're learning SAS and trying to get into clinical programming or develop ADaM data sets or TLFs, it's hard to find data to work with and hard to get SaaS for free. But with R, you can get it for free, and there's lots of infrastructure out there to kind of get familiar with this topic. So there's data packages we put together that use STTM mock data and ADaM data that you can kind of see, which we'll go over in just a minute when I do the coding. You can see this data in real play, which mimics a lot of the stuff that I interact with daily. So I think these are important packages to kind of see, and we will get into them in just a minute. All right, so next I'm going to spend maybe 10, 15 minutes just going coding with ABRoll. The first one is just going to be a very simple get started kind of example. This is on our main web page. So this is the ABRoll homepage, and we have get started, which has just a simple walkthrough of getting started. And then, and this is why I love the R packages, is then we then have all the functions we have available within ABRoll are available here. And each function does have detailed guidance on how to use the arguments. And this is the part that I really like, is it has all the examples embedded into the documentation. And these user guides, which they're quite a lot. So the documentation in ABRoll is pretty extensive, but ADaMs are pretty extensive themselves. So we have some general guides as well as some deeper dives of specific types of ADaMs, as well as some special topic guides, which I definitely encourage check out. And something that I'm always very proud of is our changelog, which is like maybe the boring side of it, but the part that I'm always the most stressed out about is because I want to make sure the changelog has exactly what we've done, updates that we've done to the existing functions, and any impacts that users might face, as well as any breaking changes that people might face in their code. So I always think a good changelog is, I think, a powerful feature of an R package. So let's get into some coding. So this is just following some of the basic get started guide that is on the ABRoll website. So I will just run this line of code real quick. And just so we all have some basic data ready to go. This data right here that I'm reading in is from the Pharmaverse SCTM package, which is available on CRAN. You can download it and have the exact same data I have here and load it in. So the first thing I'm going to do is I'm going to read in this EX dataset, and I'm going to derive a DTM variable. And you'll notice, first thing to notice is most ADaM functions, or not ADaM, but most ABRoll functions start with derive, which is, I think, very helpful if you're trying to find pieces. So there are a lot of derived functions, but you'll notice that there's a bit of a pattern. So there's some that have param and there's some that have var, and that's two of the big topics within ABRoll. Is most of our functions are about adding variables to a data set or adding records to a data set. So right now with derived vars, we're going to be adding a variable here, and it's just going to read in this ex sttdc data set. I suppress the flag that it wants to add to it, and it's going to create a new variable with this prefix to it. So very simple. So we'll run it, and I'm going to be bouncing around a little bit, so I'm sorry if that is a little jarring. I'll try to scroll in. But I just took that original EX data set and I just added this date time variable to it. So nothing very exciting because there's actually no time on this original variable here, but do know that something happened under the hood for this variable to appear. But the big part here is that the derived vars set of functions are adding the variables to the data set. So then I'm going to merge the EX ext dataset together with the ADSL dataset. ADSL, if you're not familiar with this kind of lingo, is one of the core datasets for any drug submission. It's subject level. It usually contains a lot of the information about the patients in the drug study. But with ADaM dataset creation, I find that most of the time it's joining datasets together, filtering and ordering pieces together, and then that's your end product that feeds into the tables, listings, and figures. So here we're just doing a derived vars merge, where we're going to join or merge these two data sets together. So you could use a dplyr left join to do most of this work, but we've tried to kind of provide, like I said, like a lot of times you're doing the same thing over and over again. You're joining, you're filtering, ordering things. We've just provide some simple arguments that you can fill in to do that kind of standard function calls. So here we're gonna join those data sets together. So I've overridden that original ADSL data set. We can just check out what was put on there. Oh. So that, let me make sure that happened. Sorry, my space is so small, I'm getting a little confused. Oh, right, there was only one piece added on there. Okay, so that has been merged together, and just to bring up the function documentation. So I always kind of highlight in between the function and the parentheses, and it brings up the documentation. And I just wanted to highlight, there are a lot of arguments available to derive vars merge, but you do have, you can use a subset of them to do a lot of your joining. But I did want to highlight kind of the ones that I use a lot are filter add, new vars, where you can rename variables, and ordering how you want the joined, the merge to occur with the mode. So I definitely encourage folks to read through here and just, oh, this is a good example here. So we do try to use lifecycle to kind of alert users to arguments that are being deprecated. We did make a big push in our 1.0 release to kind of identify arguments that weren't aligned with other arguments. So we try to make it the user experience more seamless through all the different functions. So you will see from time to time us deprecating functions or arguments, less functions and more arguments now, to kind of ensure the users have the same experience across every function. So very simple. We just added a simple variable using the derive vars functions, but now we're going to go into adding records to the data set. So we're going to use the VS dataset, so vital signs. And we're just going to use some dplyr functions like filter to kind of filter things down. And then, again, just using dplyr, we're going to just rename some variables. And I'm not going to show those, but I did want to showcase. So this is something that you'll typically do in an ADaM dataset. Besides adding variables, you'll add records based on information in the data set. And so here, whereas here at the top, we added derive vars DTM, we added a variable. Down here, we're going to add records. And you'll notice that the name of the function changed slightly. So these derive param computed functions, very powerful, very useful, that you can feed in information using the set values to create a new record. And we'll see that in action in just a second. So it's taking information from these two parameters and feeding it in and deriving a new analysis value for you. So we'll read that in. So our original ADVS data set had 116, and now we're at 174. And we'll bring that guy up. Oh, way too many variables to look at. So let me run this, and we'll just have a little peek at the the information. So I have my original information with the blood pressure, and I want to see my new map records created. And you can see here the derived param computed added these records based on the information that we supplied it onto this data set. So again, that's the two big concepts in ABRoll is or important concepts. There's some other stuff that's I think pretty cool in ABRoll, but I'm not going to cover that today. But is adding variables and adding records, and a lot of the adding records functions start with derived param, but there are some deviations like derived LOCF and some other ones that we're not going to see today. So next, just wanted to add a little bit more complexity to it. I'm not going to go into the details of running all the code, but within ABRoll, we have supplied a bunch of starter code or templates, we like to call them, for a lot of the data sets that you might encounter on a drug study, from con meds to EG to exposure to labs, excuse me, to ADSL. So we've put together these templates of code that you can access with just a couple clicks to get started. So here I'm going to pull up the code for ADEG. And so within ABRoll, you can see here us constructing an ADEG dataset. One thing to notice in a lot of our templates is that we haven't really made any effort to deal with metadata, which is a huge part of building ADaM datasets. Metadata can be information on, ranges for different variables, to how you derive the variable, to variable labels, to the if it's an integer or character variable. But within the templates, we've tried to keep it pretty light. So a lot of the beginning of the templates have these little tibble tribble informations for different kind of like metadata that could be applied in other ways. And if you're interested in that and how we work with metadata with ABRoll, the Pharmaverse example site that I brought up earlier has some information on that. But yeah, so you can just save that and have your own ADEG script prepared for you. They're not fit for purpose, so I definitely don't recommend that you take these and run with them for your drug study. There's probably a lot of things that you need to do to it, but the core code is all there for doing a lot of the basic derivations that you would experience on an ADaM data set, from doing your ADY variables, your start and end date time variables, to deriving specific information for different types of data sets. So you can see here. So I think that I'm going to end it there. I think the big point for the ABRoll R package is it's modular. We've really tried to build the functions to be modular, that you can use it for doing different derivations. But then if you want to use BaseR or other tidyverse packages, you can use that as well. I think the documentation that goes along with each function, I think I'm very proud that we've done a good job of documenting how the function works, but providing pretty exhaustive examples of how to use the function. I think coupled with the Pharmaverse SDTM and ADaM packages, you kind of have a way to learn a lot of the day-to-day work needed to build ADaM data sets that feed into TLFs that then go into a clinical drug trial submission. So I think that's going to be it for me. And I'm happy to answer any questions that folks might have around the package, or just things I talked about around the doing drug submissions in R and efforts around that. And thanks again for tuning in. I really appreciate it. And thanks again for Absalon for organizing.
All right. Thank you so much, Ben. So if anyone has any questions, please leave them in the chats. You can also join us on the main stage as well. If you like to ask him your questions directly, I can add you to. Okay.
And Eduardo Mangidi just posted in the in the chat about if you're interested in contributing to ABRoll. I always encourage folks that if you have some time to jump in, but did want to share, we've tried to make it easy for people wanting to contribute to the package. So we have this dummy issue for onboarding. So you can watch this YouTube video that walks you through adding a function to the package, and we'll actually probably most likely me or Eduardo will go through how to we'll give you feedback on your first function. It's very simple, but that way you have like at least a bit of experience with the expectations around ABRoll. And if you've never used like GitHub Actions, we'll also explain to you how to like deal with any issues that come up with the actions.
Awesome. And we have a question from Steven. So it's, have you further developed or plan on utilizing metadata in ADmiral, such as using the Metacore package?
So, oh, I guess Metacore is so Metacore, our package. So, Metacore is and MetaTools are two packages for working with metadata. And these are, oh, this is a read only. Here we go. So we try to keep each package kind of scoped to a purpose. So like ABRoll is really focused on the basic ADaM datasets, whereas Metacore and MetaTools, which can play a huge part, metadata can play a huge part in building the ADaM datasets. We try to keep that separated out, mainly because the two codes can blow up really quickly. So having them in scope packages, I think is much better. But Metacore and MetaTools plays very nicely with ABRoll. And if I can go to Pharmaverse examples, there are some great walkthroughs of using specification files uh, and all the tools available in MetaTools for using that metadata from the spec files to kind of build your ADaM dataset along with ABRoll. And I think a lot of the MetaTools functions kind of they take the information from the spec files and apply them to the data set. So, but yeah, no plan on building that into ABRoll, but we always think of Metacore and MetaTools working alongside ABRoll to build ADaM data sets. But yeah, thanks, Stephen.
All right, awesome. And I think Eduardo also added a comment to further expand on that point. So I have one question. So what are the main goals for Admiral in the coming months?
Oh yeah, so I think one piece, I mean, this is like under the hood information, but one piece that we wanted to push is so date time stuff is actually really complicated, and we've built a lot of, I think, powerful functions for doing date time, building date time variables in ABRoll, but some of the code is uh, complicated, and we were trying to look at ways to reduce that complexity. Uh, if you guys aren't familiar with there's things called linters that you can apply to your code that kind of checks the cyclo, I can never say this word, cyclochromatic complexity of your code. And we're right at the limit of that complexity in some of the date time functions. And we wanted to revisit that complexity and see if we can reduce it just for long-term maintainability. And one big push that we made just recently, which I think is always very helpful, and let me go to the blog. The blog was we did just try to increase the user messaging that we give to users when something goes wrong in the function. And hopefully the blog post renders. Yeah, so there's actually a nice little example of how, well, you can barely see it, but we try to improve the messaging to the user by, sometimes we didn't always pick out the function in the stack that was causing the issue, which can be confusing the error call was sometimes to a function that wasn't available to the user, which we thought could be confusing, and we wanted to kind of work on that kind of messaging about how to fix that. And then we also have supplied like information to the user to kind of pick out functions within ABRoll to use to maybe help answer that error messaging. So that was something, a big push that we just recently made, and then in the future with the error messaging, and in the future we're looking at kind of the complexity with some of our date time functions and trying to refactor that.
All right, that's really cool. Okay, so I think we can wrap it up here. Thank you so much, everyone, for staying till the end of the session. We hope you gain something out of it, and you'll be using ADmiral and contributing to it. So if you'd like to learn more about ADmiral, the link to the Pharmaverse Slack channel, the GitHub repo, the documentation is all in the chat. And of course, I will be dropping links to Epsilon's Shiny community and our newsletter if you'd like to stay up to date with the next Shiny gathering and other things, other cool stuff we're working on. So again, thank you so much for joining us, and hopefully we'll see you in the next Shiny gathering.
Thanks, everybody.
And thank you, Ben.